Now we are going to talk about immune system in detail, and let me make you aware of the topics that we are going to study. Firstly, we are going to see that how skin works as a natural line of defense in immune system, and that is non-specific. Then we talk about what lies in WBCs. This is repetitive, you must have studied it in your earlier classes, how WBCs help in the immune system. We are going to talk about interferons, and then later on we talk about acquired immunity, in which we discuss about types of cells, and in that we talk about antigens and antibodies. All right. So let's talk about these three, a short idea what this part of the body is involved in, how this part of the body is involved in the immune system, the non-specific resistance against a disease, or antigen to be specific. You know very well what an immune system is. Every organism has an inherent property of showing some sort of response to a foreign particle inside it, and that is termed as immune system. While we reach at this stage of our syllabus, we have seen that even in bacteria, we had seen that it has an immune mechanism of restriction endonucleases. It was nothing else but an immune mechanism in which the bacteria had learned how to control viruses. So you can very well imagine that human body would be so advanced in this particular system. And we talk about skin in this. Skin provides the physical barrier to the bacteria, and of course it is local defense system. It is the physical barrier and the layer of skin known as stratum corneum works against the infection of bacteria. This is to be remembered in the case of skin. Talking about WBCs, the WBCs have neutrophils. Neutrophils are a type of white blood cells which are designed in such a way so that they attack the foreign particles in the bloodstream. So WBCs also provide non-specific line of defense against the microbes and foreign particles. Apart from this, we have macrophages. Macro means huge in size, and phages means that which engulfs something. So they are going to engulf the foreign particles or the bacteria. Large sized cells present in the WBCs. How they are formed? Whenever a cell gets infected, it releases monocytes. And those monocytes, they get converted into macrophages at some stage, and then again engulfing of the foreign particle is done. What about interferons? Interferons, you have to remember, are glycoproteins. Now, what glycoproteins have to do in immune system? They are the glycoproteins which are released by infected cells. Which cells? Those which have been infected by virus. So we have virus infected cells which could release glycoproteins known as interferons, and then these cells, they invite other cells so that they can be digested or they can be eaten up, and in a way the viral cells could be killed. And once the cell have been killed, then certainly the virus will not be able to spread the infection. For that you need to recall what you had studied in the case of viral action in infecting the cells. Now we are going to talk about the acquired system. See, the parts that we have studied over here, they are showing a non-specific defense. Okay, non-specific means even if a bacteria comes, then also the cells are going to show same effect. Even if an allergen comes, then also they are going to show the same effects. Like supposedly the stratum corneum of the skin has to show resistance. It is not going to show specific effect in the case of bacteria and some other specific effect if it is a virus, or otherwise it is an allergen, because these type of immune responses are non-specific. They are not guided at a certain foreign particle. Till now I am using the term foreign particle. Now onwards I am going to use the term antigen. Now what is an antigen? Antigen is anything that can help the body produce antibody. Now these two terms you have to understand, and once you have understood these two, then we'll come to acquired immunity thing. You remember we have talked about innate immunity and acquired immunity. Innate was this one, and now we have to take into account the acquired immunity, in which you did discuss about active and passive. Now we have to see that what is an antigen, what is an antibody. Antibody is an immunoglobulin that is produced in response to an antigen. Now antigens could be of two types. One would be complete antigen and the other one would be incomplete antigen. Now the complete antigen is able to produce an antibody response against it, and the incomplete antigen attracts the neutrophils and other cells to destroy it. So we have complete antigen and we have incomplete antigen. Now this antigen could be anything. It could be any foreign particle, any living particle, bacteria, virus, or whatever you can consider, which is foreign to the body. It is coming inside the body. It is triggering an immune response, and to that specific antigen, a highly specific immunoglobulin known as antibody is released. Okay, this you have to keep in mind. Now, how many types of antibodies are there? This you have to remember. We have antibodies designated as Ig, and we have IgG, IgA, IgM, IgE and IgD. These are the types of antibodies, and they have wide range of action, and they target many types of antigens. They have a broad spectrum against the antigens. They are produced inside the body. As I told you, they are immunoglobulins, and immunoglobulin, you must have seen the word globulin coming. That means they would be nothing but proteins. While we are talking about that they are proteins, we are going to see their structure as well. You must have studied about the structure of an immunoglobulin. It is something like this. There are two light chains and two heavy chains which are joined to each other by disulfide bridges. Okay, these are the disulfide bridges which join the light chain and heavy chain. Of course, the chains are of proteins, so we have this arrangement of antibodies. Okay, the structure is known as L2H2, 2 light, 2 heavy. Okay, 2 light chains, 2 heavy chains of proteins, and they are joined by disulfide bridges. This is what an antibody looks like. Alright, now over here we have an antigen binding site. Now this is somewhat parallel to the enzyme action that we said, that there is a binding site. Antigen would come and bind over here, and then the immunoglobulin would undergo a conformational change and break down that antigen, and then afterwards the cells would come and consume it and in this way immune response would be carried out. So this you have to keep in mind, that immunoglobulin is having 2 light chains, heavy chain structure. It is of 1, 2, 3, 4, 5. These are the types of immunoglobulins which are present. These are nothing but antibodies. Now what is an antibody? It is an immunoglobulin protein which is produced inside the body against an antigen. This antigen could be of 2 types. If it is able to bring about an antibody production in the body, that is known as complete antigen. If not, it is known as incomplete antigen. So this you have to keep in mind, and now we are going to skip to the acquired immunity topic.